Undoubtedly, pineapple stands as the quintessential fruit, with its taste and freshness serving as the authentic reflection of Costa Rica's vibrant tropical culture. These reasons makes this exotic and sweet fruit, which thrives in the 57,000 hectares dedicated to its production in Costa Rica based on data presented by studies from the University of Costa Rica. Pineapple is appreciated on an international scale to the extent that 98% of national production is earmarked for export. Moreover, it holds significant economic importance due to the profits it generates for the entire country, with a total of $941.4 million in revenue for 2018, according to a study conducted by the UCR, by placing its product in American and European markets. This international presence has driven the country's economic advancement, resulting in a surge of both direct and indirect employment opportunities. If pineapple experienced an outstanding 200% increase in 2007, as reported by UNED, one can only imagine how this grown in the scale over the years. However, while pineapple production is a cornerstone of the Costa Rican economy, a dilemma hides beneath the surface, necessitating reflection. It is not without challenge to be confronted. In this case, we are faced with the quantity that the harvest period and cultivation conditions are not aligned with global demand. In response to these situations, alternatives have been implemented in the country to meet market demands, often resorting to the excessive use of agrochemical designed to expedite production and prevent pest infections. The clear example is the fungicide mancozeb, which is a complex of manganese and zinc known as ethylene bisdithiocarbamate. According to nearly half of all the pesticides used in pineapple plantations, this highly topically toxic fungicide is applied by workers in these plantations using airplanes and backpack sprayers. Mancozeb has been linked to a range of concerning health issues, from ocular irritation and allergic reaction to chronic disease such as endocrine disruption and Parkinson. Mancozeb is applied in accordance with the fruit's development stage, avoiding its use during maturation. The dosage employment is 2 kilograms of mancozeb per hectare. Extrapolating this quantity to the total area allocated to pineapple cultivation previously mentioned as 57,327 hectares result in a total of 114,654 kilograms of mancozeb used exclusively in pineapple crops. This fungicide is applied twice during the pineapple cultivation season, with an interval of 7 to 10 days, as susceptibility to fungal infections increase during this period. Also, the prevalence of mancozeb in national production is corroborated by a study of dithiocarbamate residues, the chemical family to which mancozeb belongs. In this study, tomatoes were collected from 20 farmers and 25 market vendors, and the sole dithiocarbamate detected in 47.4% of the samples was mancozeb. This demonstrates the significant prevalence of this chemical not only in pineapples but also in other horticultural and fruit products.
Furthermore, according to the CAREX Carcinogen Exposure X study, it is estimated that around 49,000 people in Costa Rica are exposed to this pesticide. An analysis carried out on 49 mancozeb applicators in pineapple fields revealed elevated levels of the hormone responsible for stimulating the thyroid, raising concerns about its impact on human health. This finding was replicated on a sample of 57 banana workers who used this fungicide, thus demonstrating the same pattern that consists of the relationship between thyroid condition and the use of this agrochemical. Through the exhaustive analysis of this fungicide, we have identified the main agent causing negative effects in the ecosystem. In this case, It is a compound resulting from the degradation of mancozeb, known as ethylene or simply ETU. This metabolite is formed as a result of hydrolysis, oxidation, and microbial degradation processes. It has been shown to have carcinogenic, teratogenic, and mutagenic properties, which generates growing concern.
In addition, health problems related to respiratory tract infections have been identified, as noted in a study by Stutler. This study shows that children and women exposed to this chemical compound may suffer chronic damage as a result of this exposure. These findings underscore the urgent need to address the environmental and public health risks associated with the use of fungicides such as mancozeb.
The degradation process of mancozeb not only triggers the release of zinc and manganese, but also creates a harmful imbalance in micro and macronutrients crucial for crop development. This situation can result in soil infertility, which has serious consequences for agriculture and the environment. However, the impact extends beyond the crop fields. Workers who handle this chemical are exposed to ethyleneuria (ETU) through the skin or inhalation, which poses serious risk to their health. But what is even more disturbing is that the general population is also exposed indirectly through the aquatic systems and soil. Pollution of aquifers has a devastating effect on wildlife, affecting aquatic animals and birds due to the application of pesticides by spraying, which allows these chemicals to travel through air. The secondary effect is this pollution includes soil degradation, which in turn leads into depreciation of homes near pineapple plantations.
Based on the analysis of this problem, we decided to create a genetic circuit capable of providing an innovative solution that would benefit our farmers. The general idea of the project is to design in silico a genetically modified E. coli. The goal is to make it capable of degrading the pollutant derived from mancozeb, the The most likely product is expected to be ethylenediamine, according to what Lee said in 2010. This compound favors plant growth in soils contaminated with zinc, which is one of the components of mancozeb. In addition, this compound can also biodegrade to other substances such as glycine, which is able to improve the mineral and psychological characteristics of leaves towards higher yield quality, according to what Zucker said in 2020. Glycine can also be degraded to other substances, which are potentially soil nutrients. With this, the project is intended to be able to cover two issues. Firstly, the contamination by the fungicide, and additionally, the damage caused by monoculture plantations in soil. This is due to the fact that when there is no variety of species, only the same nutrients are absorbed from the soil, which in this case is what the pineapple needs, causing the degradation of the soil. Therefore, the project, in addition to solving a local problem that affects several areas, is also a solution to a general problem. It is based on genetic design by selecting the characteristics of different organisms that are of interest to the targets. In the absence of an organism that naturally degrades the target molecule, one that is capable of degrading a similar substance was selected. This is how manganese peroxidase was arrived at. The molecule it naturally degrades is very similar to it, although it was not synonymous with the one it was able to synthesize. It is for this reason that several tests were carried out with computer programs. Based on different parameters, it was possible to determine that it is effective. In this case, we use a tool available online called HDoc, apart from Autodoc 4 and Swiss model, in which most of the results were satisfactory. It should also be noted that a kill switch system is in place to ensure that the organism will not leave the bioreactor, where the process will be carried out to maintain the regulations of genetically modified organisms. Using an E. coli, we generated a plasmid initiated by a lactose-inducible promoter. We added an RBS that will tell the polymerases where to start the transcription, and we are ready to generate our enzyme called mayonnaise peroxidase, who initially underwent an optimization in order to adapt it to our chassis. Thanks to its structure, our product can degrade DA2. Our enzyme has multiple binding sites, of which the number 11 will be the place where DA2 binds. To get a better response of our enzyme, we purify it under the action of a polyhistidine tail, being its case of 10 histidines, which will be produced immediately after the coding sequences of our protein. At the end of the circuit, we place an RBS again, but this time for a GFP that signals the effective production of our protein when exposed to ultraviolet light. As final part, we will have a double chain terminator that brings stability to the circuit. However, at EcoSoil, we take into consideration the importance of maintaining a safe and mishappenings-free workspace. This is why we added a biocontainment system, which is a shorter plasmid with a tetracycline inducible promoter, which when activated uses an RBS to generate an RFP reporter protein bound to a toxin called RELE and a double terminator. The existence of a red staining would give us the signal that the biocontainment system has been activated. We use the Cartagena protocol to generate policies that maintain the genetic invariability of our products, which prevents the manipulation of the genetic code of our food. However, EcoSoil uses an alternative system in which we do not handle the genetic code, and we do not release an organism into the environment that could alter our ecosystem because our enzyme is produced in the laboratory and we only use the product already harvested and properly packaged. With this, we can maintain the safety and reliability of our project. As part of the actions to inform and understand our community, we have conducted interviews with people who work with pineapple and mango sap to better understand the point of view on this problem. In addition, we have been given the task of disseminating to the Costa Rican community aspects of synthetic biology and our project through informative presentations and other various dynamics such as interviews in media, as the university newspaper and radio.
Now, we have a proposal for EcoSoil project to also develop as a bioenterprise dedicated to create innovative solutions through synthetic biology and biotechnology, particularly in the agriculture and environmental field. We have the idea of creating a gel in emulsions that contains the modified enzyme, which, as been mentioned, is able to degrade the thiocontaminant component and improve the quality of the soil when it is applied in the area of interest. The project has a clear mission to lead innovation and biotechnology applications and therefore achieve to solve environmental challenges caused by the pineapple monoculture in Costa Rica. Our commitment is to preserve sustainability and promote a cleaner and healthier future for generations to come. Our values include sustainability, innovation, responsibility, collaboration, and excellence.
We initially considered using a fries drying process. However, it is costly and energy intensive, so the emulsion is a more affordable alternative. It is easy to use and meets the needs of the local producers. To create the emulsion, first we need to extract the modified enzyme by bacteriolysis, and then we must add auxiliary substances such as surfactants and binders. So these components allow keeping the enzyme suspended in the system. It is important to clarify that the formula is theoretical and laboratory tests are required to verify this effectiveness. The main advantage of this product is its ability to improve the soil and the pineapple crop in a simple way. In addition, it has been designed in different presentations to meet the needs of growers. The target market are the farmers associated with the cannabis, and we plan to offer the product both online and at the point of sale. EcoSoil has considered financial aspects, including fixed and floating costs, and source of income such as selling the product at a wholesale and retail, partnerships with affinity companies, and announcement section in our website. So now we would like to extend an invitation to farmers, business, and the community to join our cause to promote a sustainable agriculture in Costa Rica. In order to this become a reality, we need collaboration and support. An option would be to share the project to your partners or the community in order to increase the positive impact on the environmental and the local economy. In addition, by supporting research, you can help develop even more effective and sustainable solutions in working with EcoSoil. Follow our story and EcoSoil's discoveries. Stay informed about developed and sustainable agriculture through synthetic biology, and join us on our journey towards the creation of a sustainable environment.